बुक ले जाओ। And this is the ovarian vessels. This is the isthmus fertilization happening ampulla and fundibular matter of embryos. So first of all, we start when a human female zygote is formed. The primordial germ cells are found in the yolk sac, which migrate to the ovary. There they are called the oogonia. The oogenesis will start and they will have something around 500 to 600 oogonia. They will develop into primary oocytes which will get stuck in diploid of procreate 1. At birth, there will be 1 to 2 million oocytes which is not being penetrated after that. Now at puberty, GnRH will come and it is going to stimulate the anterior receptor to release the follicular stimulation. It will stimulate the follicles and the hairs are going to get outside the primary oocytes. So the hairs will be granulosa cells and theta interna, theta externa and zona pellucida. Now as FSH increases, the follicular cells will release estrogen. As estrogen increases, at the start there will be negative feedback, but later on there will be a positive feedback and it will cause LH surge from the anterior pituitary. The LH surge is the main thing which causes the rapid follicles which form corpus luteum, and a secondary oocyte will be released into the peritoneum, which will be attracted by the femali into the fallopian tube. There will be two events: either there is pregnancy or there is no pregnancy, or either there is fertilization or no fertilization. So if there is no fertilization, the secondary oocyte will get degenerated in the next 48 hours, and the corpus luteum will remain for 7 days and keeps releasing progesterone as the progesterone representative the endometrial lining for weakness will release. But later on, as there is no exchange from the trophoblast, the progesterone levels drop and the lining will shut off during the menstruation and there is nothing no pregnancy. But if the fertilization occurs by the sperm, what happens is the embryo completes its meiosis and it will form the ovum and the zygote will be formed. It will go and deposit itself into the uterine lining, that is the maternal vesicle. And the trophoblast is now 6 sexual trophoblast, cytoplasm, and the beta and it will release its now it maintains the corpus luteum until the placenta doesn't come. Now the progesterone will be released and this is from the lining. And the entire cycle will be repeat after next month if there is no pregnancy, and if there is pregnancy, it will not repeat itself, and for 9 months we will develop the fetus into the baby and there will be conception. Yeah, the concept is very
It is covered in the tissue paper. Covered it with the tissue paper and then.
This is tissue paper.
This is from cotton. This one we have covered in the tissue paper. This model. All of it. Yes. 6, 7.
Here also some fertilization.
few months in the first eco development. So, hypothalamus secretes a specific hormone called gonadotropin releasing hormone. Which stimulates anterior pituitary to secrete luteinizing hormone, which at 14 day of menstrual cycle elevated to its peak means called research, which is responsible for the release of ovum from the ovary, which is called ovulation. And after the ovum is captured by the period fallopian tube and released the ectopic junction and educated in second metaphase. After the fertilization occurs, which marks the initiation of day one of fertilization development. And the sperm finishes with ovum to form zygote. And after at around day 4, 3 or 5 of development, zygote undergoes cleavage to form 2-cell stage, 4-cell stage, 8-cell stage, and 16-cell stage of higher. The second stage of higher is known as morula. After the morula undergoes blastocyst to form blastocyst, the cells in the blastocyst then further divides into two different cell masses, the inner cell mass and outer cell mass. The inner cell mass is called the embryoblast and outer cell mass is called the protoplasm. So this in the first year, this was occurs in the first year of development. Now the second year of development is where the actual binomial changes formation occurs. This will be explained by my friend.
Now I will discuss the actual our topic is binomial changes, which is occurring in the second year of development. So initially, as my friend told that in first year of development, there are major structures are trophoblast and inner muscle and blastocyst. Now, in trophoblast, in second year of development, the trophoblast is divided into central trophoblast and cytoplasm. Central trophoblast is not particular. These cells are disintegrated and these cytoplasms are actually well defined organisms and modern perspective. And inner muscle is divided into two, that is epiblast and hypoblast. Upper follicular layer is the epiblast cell and one tuberous cell that is the hypoblast cell. And there will be a cavity for in upper layer. That is the embryonic cavity. That is the structure of day, and when actual binomial jaundice, you see the day night. In day night, you see the well-defined binomial jaundice. That is become more prominent and slightly better. And this whole structure of binomial jaundice, then this second week of development is actually called food speed, because in this food, the major all the structure will become two different. That is two cavities will form. At first, that is yolk sac and amniotic cavity, and two layers before the layer and extracellular layer. And
what is the next? Explain. Anyone will explain? Continue.
Cytoplasm. And as you are divided into outer, there are multinucleated cells which have no distinct cellular boundaries, which is called a syncytoplasm. So after then you see that the cells in the cytoplasm get migrated. These are mitotic figures because they have the capability of division. They get migrated in the syncytoplasm, and after that they can release from the syncytoplasm into the endometrial stomach. So this much about the day 9. So after that we see the day 31. This the epiblast layer gets fully converted into a more well-defined layer, which is known as the exosome. It is known as the exome with the extra embryonic cavity. And the cytoplasm and the syncytoplasm remains intact at that stage. And then occurs this for the PVT yolks. But at the day 13 of PVT, yolks get further developed into more prominent stage, which is called the secondary yolks. And some remains, some part of the PVT yolks remains extracellular cyst there. After the day 13, in the last day there, 14 the whole, this not the stock, the whole stock gets degenerated, and the degenerated and this remains at the
refer to the main topic of my level, Jambi. So epiblast and hydroblast. And epiblast is also germinal mesoderm and non-germinal. Germination is also very much. If you see this germinal mesoderm in childhood development is there that converted into three generates ectoderm, mesoderm and endoderm, and this generate our whole human vegetation.
Explain. Nine.
So the first stage is blastocyst. This is the stage of 5 to 6 days of intrauterine life, where all the cells of the embryo they are in the periphery, and in between there is a fluid filled cavity called blastocyst. Then the second stage is the blastula or the mature blastocyst, where there is formation of inner cell mass. This is the inner cell mass, and this is the trophoblast layer. So from the inner cell mass, it proliferates, it differentiates into a flattened cell layer called hypoblast, and the rest of the inner cell mass forms the epiblast. So these epiblast and hypoblast together is called bilaminar disc. So from that bilaminar disc, the epiblast cell layer, they first proliferate and they differentiate and form an elevation called primitive strip on the 15th day of intrauterine life. And through this primitive strip, some cells of the epiblast they migrate between the epiblast and the hypoblast, and they replaces some cells of the hypoblast to form the endoderm, which is the innermost layer. Then some cells of the migrating epiblast they lies between the hypoblast and epiblast to form the mesoderm. This is the mesoderm layer, and some cells which are remaining in the epiblast cells which are remaining forms the outer cell layer, which is the ectoderm. So from these three germ layers, all the organs of our body develop. So the organs which are developed from endoderm are the epithelial layer lining of the gastrointestinal tract, except the mouth and the anus. Then the respiratory tract, then the epithelial layer of urinary bladder and urethra, then the etc. And then the organs which are developed from mesoderm are the heart, then muscles, cartilages, bones, etc. The kidneys and ureters also develop from mesoderm. Then the adrenal medulla is developed from cortex, is developed from mesoderm. Then the structures which are developed from ectoderm layer are the skin, hair, nail buds, then all the adrenal cortex, adrenal medulla, etc. So these is the gastrulation which is completed within 21 days of intrauterine life.
Good morning, respected teachers. Good morning, sir. Good morning, ma'am. I am Amri Chakravarti, and today I will be presenting my case, Group D. Today, I will be presenting our model on the formation of chorionic villi. So, this chorionic villi is a fundamental process in the development of placenta during the early days of pregnancy. So, first you will start with the zygote. So what happens after fertilization? The zygote undergoes repeat mitotic division, thus forming the two-cell ultimately the 16-cell morula. Then what happens? Water moves inside this morula and thus forms a fluid-filled cavity for the blastocyst. Ultimately it forms the blastocyst. Then it has two layers, innermost cell and outermost cell. Innermost cell forms the embryoblast and the outermost cell forms the trophoblast. This whole blastocyst still floats within the uterine cavity. Then what happens? This blastocyst starts approaching towards the endometrial lining of the uterus. Basically, the uterus has three layers from inside to outside. These are the endometrium, myometrium, and the perimetrium. Blastocyst gets implanted within the endometrium. After implantation, it starts releasing some enzymes that helps it to go deep into the endometrium. During this day, during 9th layer of development, the outer layer, which is the trophoblast layer, it starts dividing into two layers. The inner mononucleated cytotrophoblast and the outer scattered mononucleated syncytotrophoblast. This syncytotrophoblast, which releases some hydrolytic enzymes that helps to break down the endometrial lining of the uterine blood vessels. It also releases an important enzyme known as human chorionic gonadotrophin hormone, which is absolutely necessary to maintain the corpus luteum. And further, the corpus luteum maintains the cholesterol level, thereby maintaining the endometrium intact and thus it prevents the menstruation. Further, what happens during 11 and 12 day of development, there are small spaces that start to form into the syncytotrophoblast. And these spaces are called the lacunae. And these are nothing but small spaces filled with the maternal blood. And in 1 or 2 days, several lacunae will start to fuse with each other and they form the intervillous spaces. Here is a model of lacunae which is showing the cytotrophoblast and the syncytotrophoblast. Spaces formed in this lacunae called the lacunae, and the intervillous blood vessels, they are eroded by the hydrolytic enzyme released by this syncytotrophoblast only. Then what happens? Embryoblast that I have talked earlier. This embryoblast also develops into epiblast and hypoblast. And also two cavities are formed. These are two cavities, that is the embryonic cavity and the primary yolk cell. And another layer called extraembryonic mesoderm, which starts to develop between this cytotrophoblast and the embryonic cavity and the insect cavity. This is the orange thing is the extraembryonic mesoderm. Soon after its development, it starts to split and thus extraembryonic cavity or chorionic cavities. This extraembryonic mesoderm further divides into two layers. The somatoplasmic layer which lines the cytoplasm and the cytoplasmic layer which lines the yolk sac and the amniotic cavity. This somatoplasmic layer of the mesoderm is absolutely necessary, as I will talk in the later notes. Then what happens? The outer layer of the cytotrophoblast. This cytotrophoblast starts to proliferate and pushes upward along with the syncytotrophoblast and then these are known as the primary villus. The primary villus is the main inner core of cytotrophoblast and the outer core of syncytotrophoblast. This green is the syncytotrophoblast. Then what happens? We need to reach last end of the third group. Extraembryonic mesoderm I have talked earlier, especially the somatoplasmic. It also starts sending its projection into the primary villus, thus forming the secondary villus. Secondary villus is composed of from inside to outside. These are the extraembryonic mesoderm, cytotrophoblast, and syncytotrophoblast. Then what happens? The extraembryonic mesoderm, its tissue starts to differentiate and thus forms several blood vessels. These blood vessels starts to form within this extraembryonic mesoderm and thus forms the tertiary villus. We have a 3D model of this tertiary villus. Next, the tertiary villus. If we remove the blood vessels, it's only the secondary villus, showing the extraembryonic mesoderm and the cytotrophoblast and the syncytotrophoblast. Blood vessels starts to develop and it is known as tertiary villus. And these are known as intervillous spaces. In these spaces are the maternal blood vessels. They are eroded by the hydrolytic enzymes and these spaces completely filled with the blood. Now what happens? The embryoblast is connected to the trophoblast structure known as connective swab, which later develops into the umbilical cord. This umbilical cord is composed of two arteries, one way, two umbilical arteries, one umbilical way. This umbilical arteries branch of these arteries and veins. These are known as chorionic arteries or cotyledon arteries. What happens? We oxygenate and the OS material containing the blood. They starts to pull together and into the carbon dioxide and OS material helps the tertiary villi to get supported with the maternal side. This tertiary villi is supported through the cytotrophoblast with the help of a structure known as anchoring villi. This is the anchoring villi supporting the tertiary villi on the maternal side. And this whole plate is known as the chorionic plate. Chorionic plate and its extensive branching pattern, these are together known as chorionic fundosum, and they together form the placenta. Now, unfortunately, there are some diseases in which some infectious pathogens, they can cross this villus membrane, like all the three membranes that I have discussed. These infections, they are grouped together under nematode infection, like rubella virus or herpes simplex virus. They can reach this barrier and cause some severe congenital anomalies. Thank you, ma'am. This was it from my side.
Good.
What forms the placental barrier?
Such a medium, ma'am. These three layers together from outside to inside, these are the syncytiotrophoblasts, cytoplasm, and the extraembryonic mesoderm along with the endothelium of the blood vessel. They together form the placental barrier. They only allow specific, like all the hormones not there, they are not allowed. Only cortisol is one hormone that is allowed through this placental barrier.
Initially, after the formation of the preliminary drum disc, it is only disconnect structure. Now it has to be get into a proper shape so that the embryo gets into a, so that its organ develops, so that its gut tubes develop. So this is initially the ectoderm and the endoderm, only a plate-like structure. It later develop into a long after it becomes the human body. Initially, the extra-embryonic mesoderm cover the anion, and this is the ectoderm, and this is the amniotic cavity. Initially, the embryo is covered on the dorsal surface by the amniotic cavity, and this is the large egg sac. But there are two foldings. One is the longitudinal folding, and another is the lateral folding. In longitudinal folding, the embryo elongates in craniopodal direction, in which the head develops and another the tail develops. This is the showing the longitudinal folding. Initially, this is the ectoderm and this is the endoderm. Now, due to the development of the somites, the development of the brain vesicles, and the spinal cord, the head fold develops. And due to the development of the spinal cord and the somites, the caudal fold develops. And initially, the large embryo, as the large egg sac, it shrinks and it gets incorporated into the embryo, forming the gut system of the embryo. Now, initially, there were no cardiac, no other organs developed. So later, the cardiac regions, which will from the heart, the second cardiac which will from the diaphragm, so initially was in the cranial. But when this embryo elongates, it descends to the thoracic region. And the orbital membrane, which will later from the mouth or this pharynx, was initially towards the cranial. Then it is pushed by the embryo towards the ventral side. And initially, the cloaca, which is from the urogenital system of our body, was initially directed caudally, and it is also pushed ventrally. This is the allantois. Allantois from the urachus, which will be persisted as the median umbilical ligament in the embryo. And it is a connecting stop, which is connect the embryo to the mother body, which is developed from the placenta. Now, if we cut the embryo in the transverse section, we found this model. It shows the lateral folding. Initially, this plate-like structure, by the lateral folding, it will go into a tube-like structure. Now, we add a tube with a tube structure with that. Now, this is the embryonic cavity, amniotic cavity surrounding the whole embryo. Now, this will extend and grow more and covers the whole embryo. Now, the ectoderm, this is the extent growing of the ectoderm in the mesoderm. This structure we found. Now, this model is showing this is the extra-embryonic mesoderm, this is the amniotic cavity, and this is the ectoderm. Now, the ectoderm grows ventrally and fuse in the midline, so that the anterior podial is completed. And this is the mesoderm. This is the neural tube. Neural tube develops from the ectoderm. It's from the neural tube, then it forms the neural fold, then the neural tube is formed. The yellow structure is the motor cord, and this is the dorsal aorta. The mesoderm has three parts. One is the parietal mesoderm, one intermediate mesoderm, and another is the lateral mesoderm. The lateral mesoderm has again two parts. One is the visceral layer, one is the parietal layer. This parietal layer fuses along with the ectoderm so that the anterior body wall is completed. And the visceral wall fuses with the gut tube with the egg shape forming a smooth muscles of the blood. And in between them, the cavity is formed, the intraembryonic cavity, from which later the pericardial cavity, pleural cavity, endocardial cavity will be formed. And if the body, this force is unable to fuse properly in this midline, then there may be some defects which we call the ventral body wall defects. If it is unable to fuse in the passing region, the heart being superficial, we can see the heart. This condition is called ectopia cordis. And if it is unable to fuse in the lower abdominal portion, the urinary bladder may be visible. This is called extrophy bladder, the bladder extrophy. And if this is unable to fuse in the umbilical region, the intestine and liver may be superficial. This is called omphalocele. And this is the from my side.
Good.
Deficiency of a particular.
If we give more and more vitamin A, then it was going to be
Any deficiency of any particular vitamin that can cause renal defect.
Development of placenta. My name is Ikram Hussain, and along with my co-presenter, Divya Nath, will present you the different stages of development of placenta. Here you can see the embryo, human embryo at the second month of development, along with it, the human embryo at the end of the third month of gestation, and the full-term placenta. As we know, the fetal period begins from the 9th week of gestation to the end of the fetus that takes birth. During this period, extensive compartmentalization and growth of the organs and fetus take place. So for this, the placenta plays an important role in the exchange of nutrients, gases, and waste products from the maternal body and the fetal body. Here we can see the embryo at the beginning of the second month of gestation. Here we can see at the embryonic pole, the chorionic villi develops a lot, forming the chorionic fundosome. At the aembryonic pole, the chorionic villi do not develop much, which is called as the chorionic ligae. Along with it, there are spiral arteries from the maternal side, which brings the maternal blood to the fetus. Along with it, we can see the chorionic cavity and the venous return. The next will be treatment by Devana. So, 1 minute repeat to this. Now we see the second stage. So, in this model, at the end of the third month, we see the development of the intravenous spaces on the tertiary villi. This is shown here. The intravenous spaces are formed from the nuclei of sensitive tubular cells, and this is filled with maternal blood. The tertiary villi deep into these intravenous spaces, like trees sticking in blood lakes. So this is the intravenous space. These are the tertiary villi. This is the developing umbilical cord. This is the developing fetus, and this is the junctional layer, where you see the decidua, pericarditis, and neon in the colonic levy. These three layers fuse to form the junctional vein. Next we will go to the model. This is the model of the fully formed placenta, and this is a subsection of the fully formed placenta formed at the end of the fourth month. So in this model we see the decidua basalis, which is the outermost layer of the placenta on the maternal side. This forms septa that deep into the intravenous spaces, and this, as a result, we can see several divisions which are called cotyledons. These are called cotyledons, and we see this cotyledons. Then we can see at this stage the vessel circulation of the placenta is also completed, fully developed. So we see these are the chorionic vessels. These chorionic vessels which run through the chorionic plate and deep into the intravenous spaces, and these vessels carry the fetal blood and help the fetus to exchange nutrients, blood, and waste products with the maternal blood. And these vessels are connected with the umbilical vessels, the umbilical vessels, the umbilical arteries very deoxygenated blood and the umbilical veins very oxygenated blood. This is an exception. These are the umbilical veins, these are the umbilical arteries. And this is the amniotic sac. This covers the fetus and protects the fetus during pregnancy. So, this is the section. It shows the chorionic vessels sitting in the chorionic intravenous spaces and extending blood to the medullary blood. This is the basically thing.
Also, there is a clinical condition in which there is an improper implantation of the embryo at the lower end of the uterus, which is called the placental previa, which blocks the internal OS of the placenta.
So finally the placenta weighs around 500 to 600 grams and is about 25 to 30 centimeter in diameter. It is expelled from the body after the childbirth about 30 days later.
Our topic is development of interarterial. In the interarterial septum is related to the development of heart. The development of heart starts at the 5th week of intrauterine life, and it is the first organ to start working as early as 22nd year. The heart starts development in form of two endothelial tubes, which fuse to form four major structures. That is from caudal end to cranial end, that is sinus venosus, common atrium, common ventricle, and the cranial part is bulbous corpus. The part we are most concerned is the common atrium and the sinus venosus, which helps in formation of the atrium part. The most interesting part of the atrial development is from the right atrium. The right atrium is formed by the common atrium, that is the primitive part, which forms the rough area. Which the rough area is called rough area because of presence of musculi pectini, and the smooth area is formed by the sinus venosus. Ma'am, this is the rough area which is formed by the primitive atrium part, and the bluish part that we are seeing is the sinus venosus, which opens up from this portion and forms the smooth part. Now, from the development of the septum, the first structure we are seeing is the septum spurium. The septum spurium is formed by the at the cranial end of the two valves where it gets mixed up, and on the left side we have the septum primum. The septum primum goes from the cranial border of the atrium towards the AV cushion. The AV cushion forms the septum intermedium. The first structure here is not shown, but the foramen that is formed is the foramen primum. And when the foramen primum is closed up by the septum primum gets degenerated at the cranial end, forming the foramen secundum. The foramen secundum is then closed by another structure present in between, that is the septum secundum. The septum secundum and the septum primum, there is a oblique shaped gap, that is the foramen ovale. The foramen ovale relates to the structure of fossa ovalis in the adult human being. The foramen oval is patent during the entire intrauterine life. When the right atrium starts receiving deoxygenated blood, it gets closed. The defects that we see in the development of intra-arterial septum is the arterial septum defect, which causes cyanosis and due to the mixture of oxygenated and deoxygenated blood. We are also known as the Fallot's tetralogy, which occurs due to the not formation of the AV cushion, which is the major structure which helps in formation of the intra-arterial and the intraventricular septums. So the interventricular and interchamber persistence of this connection between the two chambers forms the Fallot's tetralogy. It consists of two or more types of diseases. The one is hypertrophy of right atrium, one is intraventricular septal defect, one is overriding of aorta, and one is pulmonary stenosis.
लोग को क्यों बनाने बोले हैं, जाने नहीं बोले। अरे Front, who is you? This side, come this side. H.
First is the bulbar part, which is seen here. The last of the bulbar is A for right bulbar ridge, A for left bulbar ridge, B for right bulbar ridge. This is the upper and this will further from the bulbar section of the intraventricular segment from here to here to here. Then the second part is from the membranous part, which is from intraventricular cushion, is here to the epicenter. From here from this. And last is the from the primitive epithelium is this part, muscular part, from the muscular sector of the last part of the
rotation of duodenum to form the C shape, it actually rotates to come to finally, in the adult pancreas, it actually joins to the C part of the duodenum, and the bile duct is behind that. So another thing, if we dissect the pancreas, we can actually see the duct system. So this is the duct of the dorsal bud, which actually continues at the accessory pancreatic duct, and if the major circulation of the secretion of the pancreas goes through the duct of Wilson, which is actually the duct of the ventral bud, and this is the accessory pancreatic duct, which is known as the duct of Wilson. And also this in the development of pancreas, there are common some anomalies such as the pancreas divisum and the annular pancreas. So in the pancreas divisum, as we can see, this connection doesn't form and the fusion of the different buds doesn't form. So the major, as we can see, the major part of the pancreas is this, the body and the tail part. So the major secretions go from this accessory duct of pancreas, and this is and there is double opening. This sometimes may be absent in most cases. So in the case of pancreas divisum, there are double ducts, and this doesn't actually cause many clinical presentation mostly, but sometimes it presents as acute pancreatitis. And also in the annular pancreas, this ventral bud actually doesn't come posteromedially and fuses to the pancreas dorsal bud. It actually forms a ring around the duodenum, which then appears as a duodenal obstruction sometimes. So this is my topic. And I also want to highlight another thing that everything we made this model from is recycled, and this is paper and this is bottles. This gut rotates, but this ventral bud actually wraps around the duodenum, the foregut. So it actually causes an obstruction in the passage of the duodenum, and it majorly presents as a burden of
Something is missing. Something is missing.
There are major three hormones, insulin, glucagon, and somatostatin by the iron.
Deficiency of which hormone causes diabetes mellitus?
Insulin is diabetes.
Our model is physiological definition of the body. So, during the first week of the molecular development, the arterial segment of the liver starts to develop very fast, and there is development of the liver, and there is the because of the rapid elongation of the arterial segment of the liver and the rapid growth of the liver and developing mesenteric kidneys. So the abdominal cavity is small, accumulate on this structure. So this results in the midgut lobe entering into the umbilical cord over this time. So this is called the physiological condition of the midgut and physiological umbilical cord. And during this time, the midgut undergoes rotations. So there is a total of 270 rotation, plus 90 degrees during the hibernation, and the remaining 180 degrees is during the return of the midgut into the abdominal cavity. So to better understand this, we can divide this into three stages. First 90-degree rotation
the post arterial segment of the midgut comes to the left. Now, the next 90-degree rotation. So, what happens in the next rotation is that the pre-arterial segment of the midgut comes back into the abdominal cavity. This is during the 10th week, and the herniation starts on the 6th week. So, return is on the 10th week, and during the third rotation, again 90-degree counterclockwise direction, the post-arterial segment returns back into the abdominal cavity.
During the fourth week, there are some developmental fields in the cervical region, which leads to the formation of pronephrons. The pronephrons include the prolific tubules, which append to the mesonephric duct. The pronephrons are rudimentary structures. They usually degenerate as there are no functional uses. The second structure is the mesonephrons. The mesonephrons start developing from the middle of the fourth week to the 8th week. It includes the mesonephric duct and the mesonephric tubules. The mesonephric tubules occur in the mesonephric duct. In males, the mesonephric duct remains persistent as it develops into the male genital duct system, including the ejaculatory duct, the vas deferens, and the epididymis. In females, however, it is of no functional use and it regresses. Then there is the metanephrons. The metanephrons develop from the pelvis region in the 5th week, from the 5th week. It includes the ureteric bud and the metanephric plasma. The ureteric bud extends from the mesonephric duct and leads to the formation of collecting system, which includes the collecting duct, the renal pelvis, and the minor calyx in the mesonephric. Whereas the metanephric plasma gives rise to the nephrons, which includes the proximal convoluted tubule, the distal convoluted tubule, the Bowman's capsule, and the spoke. Then the mesonephric duct terminates in the structure of the cloaca, and the urogenital sinus and cloaca. The cloaca has an extension known as the parenteros, and the anterior terminal is urogenital sinus. Also, there is an inferior part known as the primitive. Since the development of metanephrons occurs in the pelvis region, after developing into the kidney, it ascends into the lumbar region. Start. Start.
We have come to and our topic of the model is development of time. So this is the model showing the fourth development and the showing the fifth development. So the development of the cow is related to the development of the pharyngeal artery that starts in the floor of the developing mouth. And so the pharyngeal artery, which is the beginning of the metabolism in the current wall of the middle of the organ. So from the medial part of the past pharyngeal artery, there is the pulsation takes place for the direct urinary swelling. And also there is another swelling which is already developed in part that separates those swelling partially.
And on the second, third and fourth phalangeal arteries, there is development of hypothetical eminence, which is two parts. Upper part is training part and the lower part is tubercular part that ultimately disappear from the tongue or sometimes remain remnants of it in this blue color mark. And from the cranial part of the hypothetical eminence, the posterior one third of the tongue is developed, and from the caudal part, the posterior part of the tongue is developed. And from the behind the tubercular part, there is proliferation of the thyroglosseal duct, and the opening of it is shown in the upper surface of the tongue, known as foramen cecum. And this dark green color is known as the remaining part of the hypothetical eminence. And there in front, there are these two parts showing in the below. The acquired eminence, and upper part it is known as epiglottal swelling. And in the center there is a black color known as the laryngeal orifice. So the tongue, in the anterior two thirds of the tongue, it is supplied by mandibular nerve that develops from the first pharyngeal artery. The first pharyngeal artery also known as mandibular nerve. And posterior one third of the tongue is supplied by the glossopharyngeal nerve, and the posterior most part is supplied by the superior laryngeal nerve. And the taste sensation in the first anterior two thirds is by the chordatympanic branch of the facial nerve, which is the seventh nerve terminal. During swelling, it has swelling of all. It also prevent of root to phototrachea.
From the first pharyngeal arch, many other structures are also formed. Can you name some structures?
Some of the muscles of mastication also formed from the first pharyngeal arch. And the mandible vein also develops from the first pharyngeal arch.
Here also something. Then
what is the pinna of the
third nerve paralysis causes ptosis, dilatation of the pupil. Fourth nerve paralysis causes diplopia, vertical movement. Sixth nerve paralysis causes the eye is not able to apply.
One syndrome, Bernard syndrome, which is something related with ptosis, etc. Bernard syndrome.
No, start.
Very good afternoon, everyone. Respected teachers and judges. So we are Group L, and this is our model based on the histology of liver. So first of all, as we know, the liver is the largest internal organ of our body. Its main function is detoxification of blood, difference metabolism, and bile production and synthesis. So let's take a brief look at the external structure of the liver. So this one is the whole liver. This one on blue is the inferior vena cava, and this one on the right side we have the left hepatic vein, and then this one on red is the fossa for the gallbladder. This one on blue is the portal vein, and this red is the hepatic artery. Now let's take a look at the histology. So this one on the right side is the magnified section of our liver. This is a lesser magnification and this is the higher magnification. Now the liver is made up of these globules, which are basically the structural functional unit of our liver. It contains these very important structures. At the center is what we have the central vein. And from there we have the radiating sinusoids, which basically brings all the blood into the central vein. And we also have these implant cells, which basically carry all the implant functions of the liver, which includes hepatocytes, phosphorus cells, stellate cells, and then the lastly endothelial cells. Now, these hepatocytes are arranged in radiating blood-like structures. This, as you see, this does here, and then at the corners between the adjacent globules, we have this marked area near B, which is called the portal triad, and it has this influence structures of the ducts and vessels. This is the branch of the hepatic artery, and then this one is the branch of the portal vein, and this one is green is the bile duct. Now let's see how the blood is circulated in this hepatic globules. Now 80% of the blood is come from the portal vein, and then 20% of the blood come from the hepatic artery. These two bloods will combine and mix together in the sinusoids, and from there the blood will be processed in the hepatocytes like detoxifications and then different metabolism. And then after that, the detoxified blood will again go to the sinusoids and will drain into the central vein. And from the central vein, the blood will go into the from the central vein, the blood will go to the hepatic vein and into the subclavicular vein and into the inferior Benagala and into the rock. Now let's look at the bile duct system, how the bile is secreted and then is produced to the duct system and then lastly to the human bile duct. So the bile is sized and secreted from the sides, from here, and then from there the bile will be secreted to the bile nuclei, and from the bile nuclei it is transported into the canals artery. From the canals artery, the bile is transported into the intralobular duct. Intralobular duct is represented somewhere here, and then into the interlobular duct, and then into the intrahepatic duct, and into the common bile duct, and lastly into the common hepatic duct, and lastly into the common bile duct. And as we know, we have different types of cells here. This hepatocytes, stellate cells, corpus cells, endothelial cells, and damage or the improper functions of these cells will lead to certain clinical conditions like the cirrhosis of liver, the hepatitis, and then fatty liver, and so on. So that was all. Thank you so much. Then cirrhosis liver.
Going through all
all are very nice.
बारिश है मतलब बृष्टि है
बारिश हो रहा है
सर स्टूल लाके सर स्टूल
टोटल क्या नाम था? ठीक है ठीक है Then there is the metamorphosis. Metamorphosis develops from the fifth degree. It includes metamorphic plasma and the electric body. Metamorphic plasma forms the nephron.
मैंने डेवलपमेंट के लिए हम आस्ते आस्ते जो आईसी डेवलपमेंट हाँ ग्रेजुएशन सारखं होता ना मी पडतले हार्ट डेवलपमेंट ফিলিংসটা আছে এই মাসে আর ফাইনাল অগাস্টে মিউজিয়াম একটা আছে
this one is zero power until the higher power with the section of the active values. So basically the liver is composed of this active values which are the functional and physiology of the liver. It has certain sensors which perform different functions for the liver. The liver is hexagonal in shape and it has at the center it has a central vein which basically drain the blood from the cells and it has different types of cells like the hepatocytes which is the main cells, then complex cells, stellate cells and the cells. And at the purpose of these modules, and then we have the
but this participation is more important. And as a part of your academic curriculum, this is I think this is one of your last academic assignment. I hope in future also you will come up with when proceed toward the second phase or third phase in the final MBA also. You will show your keen interest in preparing all these type of models as well as using activity part. Because this type of participation when we do a lot of enthusiasm and discipline jumps up to exhibit your hidden talents. So my dear students, I wish all the best for your future life and hope you will shine one day and all the best to you one day.
And some encouraging words.
I actually joined here. First I joined here, and then I was the part say after 20 or 30 years. Starting to so I must thank this Diwali for inviting me here. All those who almost 30 years. Because after that I went to Kolkata. From there I started during the time of COVID. Anyway, all facilities were very good. And I need to go to the company to work.
Very good morning to all of you. I won't give you a waiting, you must be waiting for the results, isn't it? So, group F got the third position. And so, now, second position. And anyone guess who will C na F. Sorry. D.
सबको मिलेगा, सबको मिलेगा। सेट